Hello, my name is Harrison Ho, and today I'm going to be presenting exploring large genomic language models for metagenome binning. Metagenomics, as you know, is an important process that enables scientists to conduct microbial discoveries. It allows researchers to figure out which microorganisms are in their sample of interest. One key process in metagenomics is metagenome binning. Metagenome binning is the process of clustering contigs based on what genomes these contigs originated from. Two key statistics calculated in the middle of the process that make metagenome binning possible are one, tetranucleotide frequencies, as well as abundance. In the case where the number of samples is small and abundance information is less reliable, there becomes an even greater need to have better sequence composition representation. So can complex representations of sequence composition perform better than TNF for metagenome binning? This leads us to hypothesize whether or not genomic large language models, or LLMs, can represent species information better than TNF. There are a small handful of DNA large language models that have been published recently, each with a different tokenization strategy. First, there is the EVO model, which uses single nucleotides as tokens. Then GenSlim or GenSLM uses codons. And lastly, Genome Ocean is an unpublished DNA large language model produced by others in my group. It uses byte pair encodings, or BPEs, as its tokens. With all of these genomic large language models, our main question becomes, which genomic large language model generates better embeddings that can be used for metagenome binning? We decided to test this question by applying all three genomic LLMs to DNA fragments generated from Zymobiomics microbial community standards. This contains 10 known species. This includes 2 eukaryotic species as well as a handful of gram-positive and gram-negative bacteria species. Embeddings and TNF vector were generated. We then applied UMAP and dbScan to produce clusters. These clusters for each of the large language models, as well as TNF, are shown here. ARI was then calculated for each of these. We can see that here. We see that Genome Ocean performed well and had the highest ARI score of 0.92. Surprisingly, the Genome Ocean Foundation model performed similarly to TNF. We then wanted to evaluate how well Genome Ocean embeddings and TNF compared against phylogenetic distance, which can be seen as a ground truth. We created a second data set of DNA fragments based on 25 different species from the Genome Taxonomy database. These species span 14 different genuses. This allows us to take the phylogenetic distance information associated with each genome directly from the genome database as well. The scatter plot shown here shows how well Genome Ocean embeddings and TNF correlate with phylogenetic distance. The two metrics have very similar Spearman's correlation coefficients with phylogenetic distance, and we're unable to determine which metric is better. We also find that the Spearman correlation coefficient between TNF and Genome Ocean is quite strong at 0.79. So in summary, we find that embeddings can discriminate between contigs of different species on low complexity samples. Genome Ocean produces embeddings that represent sequence composition better than EVO or GenSLM. Genome Ocean as a foundation model captures sequence composition at least as well as TNF. And with that, I would like to thank all of those that have helped me with this project. And lastly, I would like to thank you, the audience, for listening to my presentation. Thank you.